attention for group 13. I'll start, okay?
Awesome. Hello, my name is Amari, and we are group 13. We will be presenting on induced pluripotent stem cells. As said by the group on Tuesday, there are many promises in stem cell research, such as disease modeling and regenerative medicine. However, because of the obstacles surrounding ESCs, iPSCs have shown to be a promising alternative. One recent controversial development in iPSC research is the STAP scandal. So last year, a groundbreaking study was published demonstrating a method to producing iPSCs. However, this study was eventually called into question and retracted, with one of the major contributors even committing suicide in the midst of the scandal. But before I tell you more about this, I'd like to tell you about iPSCs. So pluripotent cells have the ability to differentiate into any cell within the body. These are the precursors to the cells we have now. Certain genes, when activated, can maintain a pluripotent state. In theory, reactivating these genes could induce these cells to return to pluripotency. In 2006, Shinya Yamanaka proposed the FOS method for reactivating pluripotency genes. Yamanaka showed that cells could be reprogrammed through retroviral transduction. After beginning with 24 candidate genes, Yamanaka and his team determined that the simultaneous transduction of 4 of these genes, called reprogramming factors, were enough to make cells pluripotent. These 4 factors are c-MYC, KLF4, OCT4, and SOX2. These 4 genes encode for transcription factors, and these transcription factors cause differentiated cells to turn back into stem cells by activating pluripotency and repressing cell type specific genes. After reprogramming, transduced cells resemble embryonic cells epigenetically and otherwise, and are able to differentiate into any cell in the body. While this is super surprising and promising, there are still some problems. The lentivirus inserts itself into the cells, into our genomes, creating some random mutations. Also, one of the factors, MYC, is actually an oncogene, meaning that these can cause a cancerous tumor to start growing within the cell. Not only that, but less than 1% of the cells are successfully programmed, so developing a cell line takes a ton of money. Given that, there are still benefits to iPSCs over ESCs. ESCs are limited based on the number of eggs donated, but we can derive iPSCs from any cell in our body. The techniques are also much simpler. Somatic cell nuclear transfer takes 6 months to master, while transfection can be learned within a day. Because we can use any cell from the body, the procedure of harvesting eggs isn't necessary, and we don't need it the way ESCs do. So to tackle the problems of both of those, we have STAP. So STAP is basically like mature cells can be mechanically stressed to behave like embryonic stem cells through methods like acid baths and physical squeezing through a capillary tube. Obligata was the first to research STAP cells after she observed that some cells shrink to the size of stem cells with pressure. Her research concluded that 3 particular stressors worked: bacterial toxin, exposure to low pH, and physical squeezing. She also found that of the 25% of the cells that survived, 30% of those were converted to a pluripotent state compared to just 1% for iPSCs. However, not long after the papers were published, concerns were raised by other scientists. As seen here, those images were actually reused from her previous works. And she changed the caption that she was misrepresenting the images themselves. Also, a gel lane from an electrophoresis image was declared misleading, and several scientists tried to duplicate her results and failed. After further inquiry, it was found that her lab at Riken CDB had terrible data management. Dates and other information were missing from her notebooks, and samples were improperly labeled. This made it difficult to confirm her origin of samples and threw the authenticity of the results into question. So the papers, there were 2 of them that were in question by her, were eventually retracted by Nature, and she was found guilty of scientific misconduct. Further investigation into her doctoral thesis showed that there were signs of plagiarism in her figures. Despite these claims, Obagata said that their perception of misconduct was mistaken and that she had planned on appealing the decision. For one scientist, Sasai, he was found only responsible for failure in overseeing the project, yet because of media criticism and the move to disband Riken CDB, he committed suicide in August of last year. Despite this, Riken planned to reform their future curriculum on ethical education. The public response to the scandal was mixed with criticism at the committee's apparent lack of thoroughness. Nevertheless, the STAP scandal highlighted the many problems in the areas of publishing, especially deliberate data selection and carelessness. Journals such as Nature publicly claimed to take greater strides in pursuing quality assurance and revising procedures for publishing. So, quick recap. iPSCs are seen as an alternative method for creating pluripotent stem cells. They are used with the Yamanaka's 4 reprogramming factors, which I mentioned earlier. STAP cells are seen as a superior method because they're easier and quicker to make. However, because of fraudulent research, the feasibility of such is called into question. With that, I give you the propositions.
Okay, so the first proposition that we have regarding iPSCs in our presentation